Good afternoon. I'm audible, Arti?
Good afternoon, sir. You are audible.
Okay, good afternoon. Good afternoon.
Yes, you are audible. Just in few minutes, sir, we'll start our program.
Oh, sure. I'm just eagerly waiting for that.
Yes. Good afternoon, everyone. I welcome you to the technical session of the first IBRO-APRC Associate School on Advances in Nanoneurotherapeutics and Neurological Disorders. Today, I take this pleasure of introducing our first eminent speaker of the day, Professor Shyam Sunder Sharma. He is a professor in the Department of Pharmacology and Toxicology at National Institute of Pharmaceutical Education and Research, Naipur, Mohali, India. Before joining Naipur, he worked as a postdoctoral fellow at the University of Illinois at Chicago, USA, and he did his PhD in pharmacology at AIIMS, New Delhi, and M.Pharm from DIPSAR, New Delhi. Dr. Sharma is a fellow of Indian Pharmacological Society. His research interests include understanding the potential role of pharmacological agents in stroke, cardiovascular diseases, and diabetic complications. Dr. Sharma has published more than 150 peer-reviewed research papers, patent, book chapter, with about 7,000 citations with H index of more than 43. He has more than 20 years of teaching and research experiences, guided masters and doctorate students. Dr. Sharma has guided more than 100 masters and doctorate students, and he has delivered more than 100 invited talks at various national and international platforms. He has completed more than 20 extramural and industrial funded projects. He is a recipient of Organization for Pharmaceutical Producers of India Scientist Award, Shakuntala Amir Chand Prize, and Dr. D.N. Prasad Memorial Oration Award of Indian Council of Medical Research, CDRI Oration Award, P.P. Shaurya Kumari Prize, and Professor N.S. Dhala Award of Indian Pharmacological Society. He was an editor of Current Research and Information in Pharmaceutical Sciences for 12 years, and now he is on the editorial board of several international journals. He was a secretary at international level of Indian Pharmacological Society for 2008 to 2011. He has also organized several conferences, including Drug Discovery and Development in New Millennium Conference in 2004, in 2005, and in 2006. 40th Annual Conference of Indian Pharmacological Society in 2007, 11th Annual Conference of International Society for Heart Research in 2014, co-organizing secretary of IPSCON 2016, Stocks India 2017-2020, and also the organizing secretary of Naipur Pharmacon 2022. For this session, he is going to deliver a talk on translational challenges in the development of neurotherapeutics. So I welcome you, sir.
Yes, actually, thank you, Dr. Arti. I think before starting my talk, I would like to thank Professor G.D. Gupta, Dr.Narang, Dr.Arti, and Dr.Charan Singh actually for inviting me over here for this very wonderful workshop actually, and I think is going to deal with the nanotherapeutics, and which is the key thing like nowadays in the pharmaceutical drug discovery and development actually. So today, I think I'm going to talk about some of the challenges which are being faced while conducting the drug discovery studies by industry and by academia, actually. And I'll take you to some of the. Just a moment. Okay, let me share my slide. Okay. on this one, translational challenge in the development of neurotherapeutics. I think if you know that therapeutics developments involve particular neurotherapeutics. Actually, why I'm calling about neurotherapeutics, because if you see the last several 10 years, actually, very few CNS drugs have been discovered actually and they developed compared to the other field. Because of the lot of challenges which are being faced by drug discovery companies. And when it comes to the drug discovery of this CNS drug, you see, like initially, if we can work on this target selection, and if we can identify the cellular and genetic targets using various approach, like nowadays genomics, proteomics, and bioinformatic approach are commonly used. Then lead discovery, if we can find the lead compound actually, which can be actually one which is showing the good pharmacological activities actually, and then we have to optimize that particular lead actually to make it pharmacokinetically viable actually, and it show the very good efficacy and the less toxicity. All I think approaches, whether like in silico, structure activity relationship, all these kind of approaches are going to be being used. And when it comes to checking the preclinical pharmacology and toxicological studies, you need to do it like in the cell line model. When it comes to CNS studies, we always used like some of the neuronal culture actually, like which are commonly used for these kind of investigations. And even actually animal models actually of the CNS disease condition. Again, I would like to mention that variety of models need to be used actually for CNS disease conditions, because actually one model actually cannot suffice the changes which are being observed in the behavior of different animals. So these are also like challenges actually, just animal model is one of the challenge. And then clinical study, like all phase 1, 2, 3 clinical trials are being done actually, and you will see that many drugs actually might be withdrawn from the market because of CNS side effects. And that is why whenever CNS drugs are being developed, they are looked into whether they are having any effect on the addiction liability and all actually that need to be done. Okay. The next slide is on challenges in neurotherapeutic research. Can you see the next slide? This.
Sir, it is not visible. Second
Yeah,
slide.
that is why
Now
actually.
it is. Now it is.
Yeah,
Now
yeah.
it is moved.
I know this Google problem actually. This is nowadays being held that it do not show this kind of a mode. Yeah, maybe it will be smaller, but you can definitely see that one, that kind of thing. So now these are the challenges in the neurotherapeutic research, like complexity of the brain. Brain is one of the highly complex organ, because you would have seen that it has billions of neurons actually, and 10 times of that of other cells, supporting cells like astrocytes, glial cells, and all. They are going to be making this brain as one of the complex organ. And it's still so much research has been done actually, still we are not able to understand actually pathophysiology of cellular disease because of complexity of the brain. And if you see kind of a look, though it's just actually maybe the weight compared to heart, you see 20% of the glucose supply, the blood supplies goes to the brain actually taking, and is working throughout the day and night. And there are challenges like blood-brain barrier, because this is one of the major challenge actually which is being faced in drug discovery and development. Then there are reflex transporters actually, which uh, basically interfere with the drug entry into the brain, because most of the drug molecule can be efflux out. Patient heterogeneity, what we can see, there are disease to disease actually, there are patient variations are there. That some of the patients initially, when tacrine was discovered for Alzheimer's disease, it was found that it was though effective in subset of the patients, but some of the patients were not showing good effect. So depending on patient heterogeneities, that, because sometimes we don't know they are which stage of the disease, whether early stage or the later stage. Multiple targets, there are multiple targets for each and every disease condition. That's also another
challenge. And there are no adequate CNS disease model. Though there are several models, but none of them is ideal one. Predictive validity of the experimental efficacy data. That is why, because like if our experimental model is not good, then that is also not going to be good. And biomarker, establishing biomarker of the disease, like we have to always looking into the, like, can we have some of the biomarkers actually which can help in diagnosis in the early stage and the late stage. And then major challenge is always the abuse liability and other side effects. If a drug, any of the drug is having abuse liability, then those drug cannot be taken to the market, because later on they are going to be withdrawn. So most of the companies are looking into that thing. If you see that top selling neurotherapeutics like aripiprazole, duloxetine, glatiramir, lisdex, amphetamine, memantine, quetiapine, methylphenidate, interferon beta 1, then fingolimod, then azopiclone, and modafinil. So what you see over there actually, like most of the drug initially, like when they come up actually, like antidepressant drugs like fluoxetine, all they were like top list of CNS drug. Now you see that aripiprazole actually, which is a antipsychotic drug, duloxetine again antidepressant, has several drugs being used for nowadays for multiple sclerosis or Alzheimer's, like memantine and all that. So that way, like these are like nowadays the top selling neurotherapeutics in the world. Since I mentioned that, like what are the can be the translational challenges actually, like that I would like you to understand. So always there like various stage like T0, T1, T2, T3, and T4. Now you can see the basic science research is being done by the scientists involving the preclinical and clinical uh, studies actually on animals actually are done during this case. Then translational to human actually phase one clinical trial is being done actually for this one. And then you see the phase two and phase three clinical trial, and there's phase four clinical trial and the clinical outcome you will come to know, and then pharmacovigilance studies are being done. So always there are challenges from like bench to bedside actually, and uh, we have to look into like how we can tackle with these kind of challenges with these CNS drug. So you see that there are like always this kind of cartoons are being shown that basic and the clinical scientists don't really communicate, and that is always true. That like most of time, like we do not talk with the clinical scientists actually, which are there working on various uh, 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 clinical studies in the phase one, phase two, phase three clinical trial actually. What is there like demands of the different doctors actually, that we never look for that one. And what also like we need to look into that, where always actually there are a lot of challenges like reproducibility is not there, clinical relevance, whether what the problem you are taking is clinically relevant, uh, whether like somebody has a technical expertise for that, all those things need to be taken into consideration. So now you can see like academia do a lot of research. Okay, it's a kind of a, and most of those things that you research are not being translated to the industry. Actually, like, that is one of the challenge. So that is why like we always put like lost in translation, crossing the valley of death. So one of the disease condition I would like to explain to you is about the stroke. Why I'm taking, because like there are a lot of like translational challenges which have been faced by the various scientists working in the area of stroke actually, that we have to just look into that one. So what you can see over there, like a stroke, uh, kind of a two type, like ischemic stroke uh, and hemorrhagic stroke. And that you know that ischemic stroke and hemorrhagic stroke, there are like two of the different type of stroke. In ischemic stroke, the blood supply to the brain get reduced, and in case of hemorrhagic stroke, there is a bursting of the artery. And 80% of the cases of like ischemic strokes have been found, and you see that initially like it used to be the second leading cause of the death, but now like kind of the therapies are available. Actually now it has been shifted to the fourth leading cause of the death for the world. So here, like you can see over here, like there is a carotid artery. Actually, if there is a blockade in the carotid artery, then blood supply to the brain will reduce. And sometimes, actually, like during atrial fibrillation, some of the thrombus can enter into the brain and can also lead to like blocking of one of the major artery, actually, like which is middle cerebral artery, actually, that is being uh, 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 that is one of the common artery which is blocked in case of the stroke patients. And you can see kind of a like here kind of a plaque formations is there, and that lead to reduction in the blood supply to the brain. So these kind of symptoms actually which are being seen in the patients actually having like hemiplegia, half of their body get paralyzed, like hemiparesis, a weakness in the lower limb, hemisensory uh, deficit, visual symptoms, speech disturbance like aphasia, and neuro behavioral symptoms like thinking, planning, cognitive dysfunctions get affected. So that is why like one of the common like term which is being used for stroke is fast, FAST. Fast, like if face like means like weakness of the face, like drooping of the face to one side, their lips actually like get droop actually. And if you ask them arm to like raise, actually it will like just go like one of the arm weakness will go down. And speech is slurred speech actually, and always like time to act. This is an stroke emergency, and most of the hospital is not only in other country, even in India, like you will see like nearby hospital in our uh, Mohali, like this PGI Fortis, all of they have a stroke emergency over there. Now you see kind of feel like a stroke damage, like early stroke necrosis after stroke, and you can see kind of late necrosis. So this kind of damage actually, and once this kind of damage occur, then you cannot bring back the symptoms back to the normal. That's one of the major issue. Now if you look at the like the sequence of the event in this particular case, actually you will see that initially like there is a excitotoxicity. Like whenever there are NMDA receptor activation, that lead to opening of the like these channels actually, and a lot of calcium accumulation is there. So this calcium can activate NNOS, neuronal nitric oxide synthase, and that can lead to the activation or the synthesis of nitric oxide from L-arginine, because L-arginine is converted by NOS to citrulline with the release of nitric oxide. And this nitric oxide can, with superoxide radical, can form a peroxynitride radical. That's again, that can be damaging. And the oxidative stress and these process can also activate apoptosis process, and the neuronal death can be there. So this kind of like initially like primary damage. Secondary damage can be because of inflammation and the programmed cell death. And like always, like if you see in this slide, there are endogenous brain protection is there, but these endogenous neuroprotective agents are not enough to take care of the issue of this one. And then actually like if you'll see that stroke one actually like will be uh, regenerative changes actually can be there, that we have to see to that one.
Hello, sir. If I'm audible, then kindly unmute yourself, sir. Sharma, sir, am I audible?
Yes. I think I just.
Okay, sir.
Yeah, there was. Okay. Sorry, I think there was some issues.
It's okay, sir.
Actually, slideshow is not there because that's why like these slides are very small.
They are visible, sir. You can
Okay.
continue.
Okay,
Yeah.
so I'll tell you like the thrombolytic agents actually which are being used to restore or improve this cerebral blood flow by dissolving the thrombus, and neuroprotective therapy focus on the biochemical and metabolic consequences of ischemic brain injury that we have to see. So now actually I would like, since like this conference is dealing with nano formulations, I can tell you like recombinant plasminogen activators are being used actually, and they are being uh, developed actually for dissolving the clot. But again, actually, the same time nowadays, actually, like many labs actually they are working on developing some of these. Uh, uh, Recombinant tissue plasminogen activator and making their nano formulation like how they can be helpful. Even the neuroprotective agents, several neuroprotective agents are being developed. Even one of the neuroprotective agent like Idaravan, which has been launched actually over there actually in Japan actually for stroke, and many labs actually they are working on the Idaravan nano formulation. If we look at the like why there is a translational challenges, you see the fate of 430 drugs candidate in 1995 to 2015. Actually, about 430 drug candidates were evaluated in ischemic stroke for the period of 1995 to 2015 actually. And then actually like 300 drug candidates were discontinued, and about 70 are undergoing preclinical trial. So now you can see, and 40 in the various phase of clinical trial, only 19 have reached the market actually. So this is like related with some of the agents actually, but if you see the US FDA, they have only approved actually like tissue plasminogen activator as a treatment for this particular condition. Like drugs like nimodipine, NXY059, Trilajit, clomethazole, MK801. Then this is basically MK801 is digoxin, eptiganil, CGS19755, JK200775, and then YM872 actually. And the translational challenges, uh, uh, why? Because like these are the drug reached clinical trial, and then translational challenges are was the one actually like which is playing important role. Like we have to understand what are the challenges are there in drug discovery and development. So now I think this gap actually what is there like from uh, lab to the bedside actually, that that gap actually we have to try to solve this particular problem actually like with this one. So one of the always the challenge for this is basically like target validation actually. Why? Because target validation that most of time like whether the drug is reaching to the site of action actually, that is the case actually. uh, uh, so that's why like I think nano technology can play a very important role. Like can we develop kind of a nano formulations actually so that we can deliver at the site of action, And the drug release can be controlled by many ways, because nano formulations nowadays actually can release the drug at the site of action actually. And many success has been seen in case of anti-cancer drug delivery to the tumor. Actually, we have seen like drug can be delivered to the tumor. Similarly, like can the drug be delivered over there? Actually, that we have to look into the consideration. The role of target in pathophysiology is the molecular target up or down regulation following ischemia. Is drug interact directly with the molecular target, or the observed effects are indirect? How selective is the drug for molecular target, and does the drug modulate rodent and human form of molecular target? So we'll have to see that whether like always the challenge is that blood-brain barrier. You can see over here this blood capillary actually, and you can see the astrocyte foot over there, and then neuron actually, and how like the supply of different nutrients or different drug to the brain is there. So this brain microcapillaries, you can see over there, like this the lumen, and then astrocytes foot is there for involving the blood supply. And always like what we have seen, that there are tight junctions. These kind of junctions are there actually, which can be like can be modulated actually, so that some of the drug molecule can cross blood-brain barrier. Even the many people actually they are using various Trojan horse technologies actually for this kind of activity. So now you can see that for stroke, it's not like just neuron or the targets. Even in the brain, like brain, each and every like neurons are the target actually. Like depolarization, cell swelling, calcium overload, excitotoxicity, mitochondrial depolarization, free radical, DNA damage, necrosis, program cell death, microglia, astrocytes, oligodendrocytes, basement membrane, extracellular space, immune cells, endothelium, parasite. All of them can be the target for this one. And as I mentioned, like the multiple target of the stroke, like initially, like it was thought that release of glutamate from the neuron actually that is going to lead to the neuronal death. But nowadays, like various downstream pathways has been looked into it actually, and it has been looked actually whether drug targeting to those downstream pathways can be effective or not. Various like inflammation, apoptosis, DNA damage, they have been considered to be the target for this one. And in addition, like inadequacy of the experiment model, like the model what we have used, like in vitro model, in vivo models, actually, that can be taken into the consideration. And can this in vitro model be adopted to high throughput? Difficult to ascertain many of the phenotypic characteristic in vitro model, like behavioral space, and in vivo model like provide best possible in vivo measure of functional endpoint. So these models rely more on the molecular and the pathophysiological this one. And many labs actually, like we have seen many studies are even being done in ISF College of Pharmacy on the brain CNS disease models actually. So they also, like many of the faculty member, they're doing very good work actually in the area of the neuroscience actually, because I'm following their work, like even like Dr. Arti, then some share Singh and all. They're doing wonderful work on various CNS disease conditions. If you see the stroke model, like we have in vitro models and in vivo model, like in vitro models, we have cell culture models, and the brain slices actually can also be used. In vivo model, like focal cerebral ischemia models, and its different variants are being used. Then we have the global ischemia model, like BCC occlusion or four vessel occlusion models, which are commonly used for stroke. What I would like to mention that, like some time back, because of translational failure, uh, one of the recommendations actually, like one of the committee was made, it was STARE, like Stroke Treatment Academic Industry Roundtable. Like both academia and the industry came together and they developed these guidelines, like rodent model, improve blinding and randomization, perform behavioral outcome combination histology, independent replication, at least two laboratory. So they indicated like if suppose you have shown neuroprotective activity of in some model, I think it should not also be confirmed by the other lab actually independently. And we should do like study in different animal model. primate study, like extension of the promising studies in rodent. Drug administration, like appropriate route must be considered, and optimal CNS level of experimental compounds determined. Then toxicological study need to be performed, and careful dose response studies need to be done. Determining therapeutic window critical for appropriate clinical trial design. So what is required basically, like you need studies in different models actually uh, that need to be done. Dose response studies to be done. Therapeutic time window studies need to be done. Non-human primate studies also need to be done. So I think those who are working in the area of stroke, they must read these STAIR guidelines. Actually, they are very good actually for the researchers. Then we have translational pharmacokinetics actually. So that also another area nowadays is being done. The translation of preclinical to clinical pharmacokinetics is a discipline that traditionally overlooked and is likely to be a key factor responsible for failure of clinical trial. So we have to see whether the drug is reaching to the brain actually. So that I think estimation of the drug level in the brain and at the site of action is another way that we have to look how the drug distribution is there. That's also looked into. As I mentioned earlier, like can we look into the biomarkers actually? Biomarker for pharmacodynamic biomarkers as well as some of the pharmacokinetic and the drug distribution markers that we have to do. Clinical development, we have to look into all phases of clinical development, like whether how they are basically ethical issues are being involved or not, whether risk benefit ratio analysis has been done or not actually, that we have to look into it. So as I mentioned earlier, that we have a stroke research laboratories, and it is equipped with like animal video tracking system, computerized physiological monitoring system, gas anesthesia system, blood gas analyzer, microdialysis, cell culture facility, and gel electrophoresis and the brain matrix image analysis software. So if you see that this kind of a commonly used model, like if any one of you would like to develop in your laboratory, you can use that one. This is a rat model in which actually like external carotid artery and internal carotid arteries are exposed, and through this one, if you can see, this is a common carotid artery, and this is external carotid artery. Through that actually like you just pass one nylon filament actually, silicon coated actually, and which goes and block MCA. And at this particular point, actually, you also record blood flow actually by putting a laser Doppler blood flow probe at the area of like cerebrum actually. And then you see that actually like in this particular graph actually, there's a reduction that the moment you block this middle cerebral artery, blood flow goes down. And after 2 hours of blockade, actually now it has come back to the same stage. So this blockade for 2 hours can induce a lot of significant damage to the brain, and that brain infections can be looked at it actually. And simultaneously, you need to measure all physiological parameters actually like that need to be done. High throughput scanning, like sometimes like we can also simultaneously measure their blood pressure changes actually using non-invasive blood pressure monitoring system. Telemetry studies can also be done nowadays, like you implant your radio telemetry device actually, and then you can record all the parameters actually when all these kind of studies are being done. So these kind of infarctions actually you can see that kind of this white pale area that indicating the infarction, and the red area which is indicating that there's a no damage. So this kind of infarctions, like if you're blocking the middle cerebral artery, you can get this kind of problem. Neurological function tests, like no neurological functions, limping to the left, circling to the left on putting the tail, spontaneous circulation, having depressed level of consciousness. All like there are several neurological scales are there which can be used for these kind of studies. Mechanistic studies can be done like oxidative stress, apoptosis, and many more depending on your requirement. Immunohistochemistry, then you can do Western blotting of different protein. Uh, So some of study like we did actually like using free radical scavengers, polyribose polymerase, and mitogen activated protein kinase, and we targeted actually these uh, downstream signaling pathways, and we looked into whether they have any effectiveness or not. So this pathway we did actually proxenated PAR pathway, and we have seen that overactivation of like this PARP actually can lead to the damage, actually DNA damage, and that can be. So this proxynitrate or oxidative stress can activate PARP actually, and that leading to overactivation of the polyribose can lead to the neuronal injury. Various PARP and the proxynitrate scavenger we have used like FET-MPYP, FET-PPS. We also use PARP inhibitor like NU105, nicotinamide, thiamanobenzamide, isoquinolinediol, and 4-ANI. So different actually like agents we use to prove that whether they are effective or not. In addition, we also looked into whether PPR gamma play important role in stroke or not. So we took like pioglitazone and the GW1929, which is a selective PPR gamma agonist, which is a non-thiazolidine dione actually. Like. So there, like you can see, GW1929 actually like showed very good if protection at the dose of 10 mg per kg actually, and which was showing in the neurological score as well as the infarct volume actually there was a changes. And even the blood flow, like there were no significant effect of these PPR gamma agonists on the blood flow alteration. Inflammation was reduced, and even the apoptotic DNA fragmentation, you can see this DNA fragmentation, such like so much of this fluorescent cells actually indicating apoptosis is there, which are reduced by pioglitazone and the GW1929 treatment in both cortical as well as the subcortical area. Then quantification was done, and we found very good effect actually. For translational like stairs recommendation, they indicate that it's not only in one model. You need to conduct that in the different model. So what we use, like we have used like bilateral common criteria, artery occlusion, like both the arteries, actually, like you can see in the gerbil. This is a like animal gerbil. If you block actually two arteries, actually, then you can induce the condition of uh, global cerebral ischemia, because in these animals circle of Willis is not developed actually, and there's no need for this particularly like um, vertebral artery blockade actually. Just blocking these two arteries, common carotid artery, can induce a similar kind of effect. This kind of experimental design we had actually like up to 96 hours, various neurological parameter was measured actually, and then drug treatment were given at different time, 0 hour, 4 hour, 24 hour, and then all the parameters, neurological function test and biochemical parameter were measured. So we used GW1929, and we found that it had got a effect actually, like we have checked in the MCU model. Now we checked also in this particular model, and we found like very good effect with this particular agent. Hippocampal damage was also, like you can see, kind of a damage over here. It was reversed actually with the treatment. Apoptotic damage actually was also like taken care actually, like you can see over here, like this was reduced. Then inflammation was reduced actually, you can see find actually like there is a reduction in the inflammation. And then we also like as I mentioned, like these recommendations indicate that you have to have a these kind of investigations actually. Okay, I think these are repeated some. Yeah. Okay. So now I think since actually this was the conference like related with the nano formulation, so I just added some of the slides. Though we are not worked on actually like these nano formulation, but we have worked on the nanocurcumin, and we investigated the nanocurcumin effect in the different disease model, like diabetic neuropathy and neuropathic pain model. We have some of the study with Dr.Sarasija, Suresh, who is also one of the speaker in this one. So they're various like polymeric nanoparticle, liposome, nanoemulsion, polymeric micelles, dendrimer, all sort of like studies are being looked into actually, like whether they can be effective or not. As I mentioned, the recombinant tissue platelet activator, which is a thrombolytic commonly used actually. The different actually nano formulations actually have been tried preclinically, and what they found actually like they increase half-life, decrease the hemorrhage, actually improve recanalization and target to the thrombus. So this kind of like one of the paper published in the Pharmaceutics in 2021 actually that's indicating like thrombolytics like tissue plasma activator have play can play important role. Even like the release like triggering control release can be done, like it has been seen that internal trigger release like pH, enzyme, temperature can control. External trigger like magnetic field, ultrasound or phototherapy can also be used for triggering the release of these drugs actually which are being like recombinant tissue plasminogen activated. So there's a list of like several drugs actually or agent which have been tried. Some of the T7 conjugated liposome, like this is one of the peptide conjugated so that it can cross blood-brain barrier. Brain-derived like fibroblast growth factor has been tried actually. MCA model. Then tensinon has been tried. Then glyburide, edaravone, like this is the drug actually which is being used for stroke that has also been nanoparticle has been made. L-arginine also has been tried actually, and you can see the curcumin has been tried, dexamethasone has been tried, several agents have been tried. In addition, actually, I would like to just mention like I think those who are working in the area of nano formulation, they must visit these guidelines like guideline for evaluation of nano formulation in India. And I think like today's morning Professor Y.K. Gupta would have told you he has inaugurated the conference, and he is the one actually instrumental in making these guidelines because he is the president of this society for nanomedicine. So he is one of the main uh, leader or editor, I'll say, in making these guidelines actually. So they also indicate like animal pharmacological data need to be done actually, like, and you need to prove like therapeutic efficacy, study of off-target effect, peripheral pharmacokinetic, bioavailability need to be done. Animal toxicological studies as per the Schedule Y that need to be done for nanoparticle. All like GLP, like is mentioned, I think last time, just last month, they ICF Moga has ICF Moga has organized one conference on GLP. So that also need to be part of your toxicity studies that need to be done for nano formulation. And there are various other requirements actually nanopharmaceuticals actually they need to take care of safety pharmacology uh, uh, like kind of a uh, core battery safety pharmacological studies need to be done, then follow-up safety pharmacological studies need to be done, and then supplemental safety pharmacological studies need to be done for these nanopharmaceuticals because making a nanopharm changing the formulation can change their distribution, kinetics, levels, which can also induce toxicity. Even the clinical trials, actually, like these guidelines also mention about how the clinical study design should be there for this one, and what kind of biomarker can you use actually for this one. Clinical pharmacology, like phase 1, phase 2, phase 3 investigations can be done actually with this one. Pharmacovigilance, even like he also like mentioned that for all the nano formulation which are available in the market actually we must conduct actually pharmacovigilance studies so that we can come to know the safety data from clinical development. With this, I would like to just summarize that from like this bench to bedside, actually, like from like preclinical to clinical. These are the challenges like target validation is a challenge, biomarker discovery, ethical challenges, clinical development, experimental model, and we how we can actually like have various modification in the formulation so that it can reach to the target site actually. With this, I would like to thank like funding agencies, like we have a funding from Ministry of Chemicals and Fertilizer, DST, CSIR, Midwestern University, and my students. They are working day and night for in this kind of areas actually, and they are like always uh, uh, the source of like inspiration because they get actually like they are doing very good work actually and publishing in very good journals, and most of them I think many of them out of this, they have already gone abroad actually, like and many in the verge of like going abroad. Like this like had gone to San Francisco like recently. He was a postdoc doing postdoc over there actually. With this, actually, I would like to thank all of you actually for patiently listening. And if you have any question, I will definitely would like to discuss with you.
Thank you very much, sir. Now the session is open for discussion. We can quickly take one or two questions from the participant. If anyone want to ask, you can unmute yourself.
Hello, doc. Hello, sir.
Hi, yes.
This is Mohali. This is Mohali. Am I audible now?
Hi, yes, please.
Yes. Thank you so much, sir, for sharing your thoughts on like drug discovery and stroke and finding some nanotherapeutics. So my simple question is about the stroke pathology. So is it really possible? Is it to reverse the phenotypic output in stroke patients when we talk about the real scenario? For example, we can prevent the damage, but can we actually reverse the behavior in the stroke patients by using any kind of therapeutics?
Yeah, I think like when it comes to like a stroke, you know that it is kind of a half the body of these patients actually. Some of the patient get paralyzed actually. So always like if we can look into the regenerative therapy, and there a lot of efforts has been done actually by like it's not by Indian researcher, even by throughout the world. They are looking into like can we have regenerative therapies actually, some of the growth factors actually like people are trying actually can it be reversed, though like our body has also got a like always the brain has a capability also to get it change some of the reverse, because I've seen personally like many stroke patients, like one of the patient actually he got lacunary stroke actually, and only like what I seen like his speech was slurred actually, and I seen like after 6 months or so actually like it just completely recovered, and that indicate like I think our endogenous neuroprotection also play a very important role over there actually, but like I think but maybe that might be the minor case, but if it's a long, if it's a kind of a paralysis, then regenerative therapy can play a very important role, and we should look into development of the regenerative therapy for these patients actually, because otherwise like a lot of like hospitalization cost and taking care actually a lot of burden on the family members because they have to take care of these stroke patients. It's
Exactly.
not that easy actually. So I think
Exactly.
if you are planning to work in the area of stroke, I think you can look into development of the regenerative therapy actually for that one.
Okay, in continuation to this question, my second question is about what is the more practical approach, either to modulate the drug for the specific target or using the brain architecture itself. Since you have mentioned that these tight junction proteins, we can modulate their expression and we can lead to the opening of the blood-brain barrier at specific points in specific regions, and we can like reverse the process. So which is the more practical approach, either the modulation of drug or the brain?
No. Actually, like again, actually, I'll tell you like most of the disease conditions and brain, this blood-brain barrier is always impaired. If you take the condition like Parkinson disease, if you take actually Alzheimer's or stroke, because already actually because of the disease condition, inflammation is there. There is a always actually blood-brain barrier impairment is there. Okay.
Can we use that window to deliver the drug?
Yeah, so that is why I'm coming to your question actually. The one thing is always like if you modify a drug actually like because you can allow that drug to enter into the brain actually, that's good. But again actually like if you can develop kind of a delivery system which can deliver the drug at the site of action, because that is very important. Like suppose there is a hippocampal damage, so if you have you can design some strategy like you can deliver the drug to the hippocampus, not to the other areas, because otherwise like it can affect other areas too actually. So that would be better. I think both approach we have to use, because like you would have seen in the nano formulation, if they are not able to be delivered to the brain for brain tumor, then actually you have to always open this blood-brain barrier using osmotic agent or something, or maybe you inject bradykinin. So nowadays, actually, like it is there, like this approach is being used, short injection of bradykinin is used before administration of the anti-cancer drug. for the brain tumor. So I think like you can use the both approach depending on case to case basis actually.
Okay.
But actually making the new formulation and I think delivery to the brain actually always a challenge actually, and that is why like we till date actually we are not having very good treatment for stroke patient actually. Only we have recombinant tissue plasminogen activator. That's all. Okay. When it comes to neuroprotective agents, there are one or two neuroprotective agents, but you see like FDA, US FDA not approved Idarawan. Idarawan is only approved in case of Japan. Okay. So that is why many scientists they are working on like developing nano formulation for Idarawan, whether it can show the better effect or not. That's why I just listed like Idarawan nano formulation. People have looked into it. So I think there are a lot of scope. I feel like I think if drugs are not able to show the effect, maybe the formulation change can increase the pharmacokinetic, increase the delivery at the site of action, can play a very important role over there.
Thank you so much, sir.
Thank you, sir. Thank you, Mohit.
Thank you very much, sir. Anyone else want to ask or discuss anything? Okay. So thank you very much, sir, for providing us the valuable insight on various translational challenges in the development of neurotherapeutics. So we really appreciate your time and effort, and we really want to thank you once again for giving us your valuable time instead of having ill health. So thank you very much, sir.
Thank you. Thank you. Thank you. Thank you. Okay.
Thank you very much, sir.
Thank you, sir. Thank you. Thank you, Professor Dr. Gupta. Actually.
Welcome. Thank you very
Thank you
much.
very much. Thank you.
Now it's time to move to the next lecture of the session. Before we start, just I need just few minutes for just to confirm the next speaker. So very good afternoon, one and all. Sorry for a bit delay. So now we have with us the second speaker of our technical session, day one. So it is now my great pleasure to introduce to you our next eminent speaker of the day, Dr. Sarasija Suresh. So Dr. Sarasija, she obtained her degree B.Pharm M.Pharm from Government College of Pharmacy, Bangalore, and she did his PhD from College of Pharmaceutical Sciences, Manipal. She has 28 years of teaching and research experience in Al-Amin College of Pharmacy, Bangalore, also from NIPO Mohali and MS Ramaya University of Applied Sciences. Presently, Dr. Sarasija is a project director at Institute for Drug Delivery and Biomedical Research, Bangalore, and also she is a founder of startup company RGV Research and Innovation Private Limited for research in safe and efficacious medicine. Dr. Sarasija is a recipient of several national and international fellowships and awards, including DAD from German Academic Exchange Fellowship, Career Award for Young Teachers from AICTE, Wellcome Trust Fellowship, Stars Award for Excellence in Teaching and Research, and Woman of the Year by Association of Pharmaceutical Teachers of India in 2015. She has 7 patents granted, including 1 PCT, and has published above 100 research papers and presentation with 7 Best Paper Awards in conferences and journals. She has published 2 books in pharmaceutical engineering and several book chapters on nanotechnology. She has been working for over 2 decades in drug delivery research with funding from CSIR, AICTE, DST, DBT, Wellcome Trust, and German Academic Research, DAT. So I welcome you, ma'am, and today she is going to discuss on potentials of nanocurcumin for optimization of Alzheimer's therapy. So I welcome.
Good afternoon, one and all. Thank you, Dr. Arti, for the kind introduction.
Welcome, madam.
Thank you so much, sir. Thank you. So it gives me immense pleasure to be here. So first of all, I would like to thank the organizers, again, Dr. Arti, Dr. Charan, for giving me this opportunity. And it is a real joy to see Dr. Sharma and to listen to his lecture. And we were collaborators, as he had been remarking, on nanocurcumin therapies, the efficacy studies. So today my talk is on potential of nanocurcumin in optimizing Alzheimer's disease therapy. See, basically, as a pharmaceutical technologist, we have been looking at different methods to increase the bioavailability of drugs. And we have been looking at small molecules, and among the natural products, we have done a lot of work on curcumin. So we are trying to look at its various efficacies and also try to look at its challenges. So one of the technologies that we developed with curcumin was a commercially viable technology. And this technology, we started from a natural product and we were able to translate it into small molecules, beginning with, that is, we had rifampicin, and then we have worked on a number of anti-cancer molecules. So with rifampicin, Dr. Charan was the one who worked on it. We got a patent, and nanocurcumin is also like a patented technology. So I would like to go through my talk under these headings. So first, I would like to briefly look at Alzheimer's disease, then look at curcumin's potential in treating Alzheimer's disease, and then we will look at the challenges of curcumin in therapy, and then look at curcumin nanoformulations, and then finally we will conclude. So here, as you all know, Alzheimer's disease is one of the most common prevalent neurodegenerative disease, and it is mainly seen in elderly, where there is progressive deterioration and loss of the brain cells. And with the aging population, there is marked increase in the number of neurodegenerative diseases. So this could be because of either that the lifestyle, stress, or environmental challenges. They are working on this genetic or congenital situation that already exists. And among all the Alzheimer's disease, we have among the neurodegenerative disease, we have Alzheimer's disease as a most prominent and common disease, and which is expected to actually double every 20 years. Now, if you see the number of people suffering from the disease, you will see that presently it is about, say, 50 million, and it is expected to become 131.5 million in 2050. So it is developing the disease progression, and the number of people suffering from Alzheimer's disease is increasing at a great pace. Now, if you look at the definition of Alzheimer's, we can, it was Alois Alzheimer who first defined AD as senile dementia, that is age-related progressive deterioration of neurons, leading to cognitive impairment and memory loss. This happens because there is deposition of, that is beta-amyloid plaques, beta-amyloid protein that forms plaque extracellularly, and internally there are tau containing neurofibrillary tangles. So the tau protein, it forms tangles within the neurons. So this slowly, generally what happens is we have autophagy, a mechanism for removing this the amyloid and tau proteins. But what happens in Alzheimer's disease is there is increased production of both the proteins. So when there is increased production, it affects the autophagy mechanism. So there is dysfunction of the autophagy mechanism. So that means, in other words, there is buildup of this amyloid plaques and neurofibrillary tangles within the neurons. So when there is buildup, this causes, though autophagy increases, this causes release of a number of the free radicals, a number of inflammatory cytokines, a number of chemicals which bring about neuroinflammation. So there are a number of pathways that get again dysregulated, and we have actually all of them sort of seem to converge at activation of NF-kappa-B. Now, this NF-kappa-B is present in the cytoplasm, as we know, and it is a transcription factor. So, because of the buildup of this free radicals, this gets activated. Actually, NF-kappa-B is, as we know, it is in an inactive form. So this gets activated by TNF-alpha, and this NF-kappa-B goes to the nucleus, and there it brings about transcription of a number of genes. So all these genes, they are all, that is pro-inflammatory genes. So where actually the mechanism is, it is trying to protect the body. The immune cells get activated. All that result in, that is the buildup of free radicals, a lot of inflammatory molecules which actually destroy the neurons. So the buildup of free radicals, it causes mitochondrial dysfunction. So finally, there is death of neurons. So what happens is that this again affects the blood-brain barrier. And so finally, we find that there is a leaky blood-brain barrier. So again, you have immune cells entering into the brain, the transport of the cells, that is, since the structure of the blood-brain, the capillary network is affected, what happens is we have a number of molecules entering into the brain, and as a result, the brain function is affected. So what is the present treatment? So present treatment is we are looking at trying to see that acetylcholine is not destroyed, because amyloid plaques, amyloid as such, it causes a decrease in the production of acetylcholine. Acetylcholine, we know, is a neurotransmitter, and its production is decreased. So what we are trying to do is we are trying to give drugs which will block cholinesterase. We know that cholinesterase is the enzyme which brings about the hydrolysis of acetylcholine. So we are actually looking at the end of that is a damage that has been created. But what would be ideally better is if we could, that is, see that the amyloid plaques are not formed? Or can we decrease, that is, are we able to reduce neuroinflammation? Because neuroinflammation hastens the disease progression. So, or can we protect the mitochondria? That would be our aim, instead of looking at the end of the pathway. So with this, let us look at curcumin. So curcumin, we know that it is the active principle of turmeric. So turmeric, we know that it has a number of beneficial effects. It has been used in traditional medicine, in Ayurveda, for inflammation, for that is anemia, for diabetes, for that is as an anti-infective, antibacterial, antifungal. It has a plethora of benefits. Now, the active principle of turmeric is actually curcuminoids. That means we are having three moieties. So first is curcumin, which we all know, and then we have, which is about 77 to 80%. Then we have about 15% of dimethoxycurcumin. Then we have a small amount, about 5% of bis-dimethoxycurcumin. Now, if you look at the structure, you can see that dimethoxy, there is this methoxy group is not present. In bis-dimethoxy, both the methoxy groups are not present. So if you see the molecule of curcumin, it's actually a very small molecule. It is having a molecular weight of about 360 Daltons. And it is having, if you see, two phenyl groups, two diaryl groups. And you are having a linker molecule, a heptadiene-dione. And here you can see that, so this linker connects these two phenyl groups. So with this, this chemical structure of curcumin is responsible for its plethora of activities. So let us see what it is. So before that, let us look at the recent work that has been done, which indicates that it has a number of benefits. So next slide, please. The biological activities of curcumin has shown that sorry for that interruption. See, a lot of work has been done from last 2 decades that have shown that curcumin exhibits anti-inflammatory, antioxidant, chemopreventive, chemotherapeutic activities. Recently, there have been studies which show that curcumin exhibit neuroprotective effects in neurodegenerative diseases. So this can be attributed to the structure of curcumin, for which we have all these activities. All these activities are because of the unique structure that we see in curcumin. So if you see the molecule, the linker molecule, it provides flexibility to curcumin. And because of that, it is able to directly bind to targets such as NF-kappa B. And also directly it can combine with amyloid and it prevents its aggregates. And this is one way that curcumin acts. Another way is it combines with targets. Either it upregulates or downregulates targets, which again modulate a number of biochemical pathways. So either it is directly binding to amyloid, or it is binding to NF-kappa B, or it is binding to targets that play a role in either upregulating or downregulating the biochemical pathways. So these pathways, especially anti-inflammatory and antioxidant pathways. So with this, thus we can see that curcumin, in case of NF-kappa B, it is able to actually, that is prevent, it is able to bind in such a way that NF-kappa B does not translocate to the nucleus and produce a number of its, cascade of pathways. It is also able to combine with NF, that is tumor necrosis factor. It is able to combine with lipooxygenase, then reactive oxygen species. It is able to combine with xanthine oxidase, nitric oxide. That means that is a plethora of biochemicals that are causing the damage that we see in case of AD. So thus, with this, we can conclude that curcumin, yes, it is a very good molecule. It can be used for treating Alzheimer's disease. But there are a number of challenges which actually prevent it from being in therapy. See, though we have done a lot of work, we have seen that it has anti-cancer properties, it is having chemopreventive properties, but we don't see curcumin in the therapy. It is not still a drug. So the main reason is that it is having poor oral bioavailability. The bioavailability is only 1%. And this is because of its poor water solubility. Along with water solubility, it is having extensive pre-systemic and systemic, it undergoes extensive pre-systemic and systemic metabolism. So it undergoes degradation in the intestinal wall. It also undergoes degradation in the liver. So this metabolism is one of the reasons, is a reason for its poor oral bioavailability. And this metabolism is by sulfation and glucuronidation, and both of them cause a small amount of curcumin to be absorbed. And it has been seen that in early phase 1 studies, curcumin blood levels were detected, very low levels were detected with even high doses of 10 to 12 grams per day. So we know that currently in our diet, we use a small amount. They have used an amount of 10 to 12 grams, even then small amount could be detected. So thus we can say that it is the poor bioavailability of curcumin that is responsible for its not being really accepted in therapy. Along with that, it is having poor stability. It is having poor light stability. That is also one of the reasons. Next slide, please. So we have been looking at various strategies to enhance curcumin bioavailability. So one, we looked at nanoformulations to increase so we looked at one method was nanoformulation to increase curcumin bioavailability, and we evaluated extensively lipid nanoformulation approach, and then also we looked at cyclodextrin complexes. So in lipid nanoformulation, we basically tried to increase its oral bioavailability by preventing the metabolism that takes place in the intestine and in the liver. Whereas in cyclodextrin complex, we try to increase the water solubility. And I'm sure you're aware that cyclodextrin is one of the easiest way to increase the solubility of drugs, solubility of flavors. It is extensively used even in the food industry. The second method that we tried is to conjugate curcumin with number of drug molecules. We conjugated it with diclofenac. We conjugated it with metformin. And we were able to actually combine the advantages of curcumin with diclofenac, that is prevent its that is a side effects that is associated with diclofenac. Then we tried co-administration of drugs to reduce the toxicity of the drugs by incorporating curcumin. So we were able to reduce the toxicity of INH, that is the liver toxicity that is associated with isoniazid by curcumin. So these were the different strategies that we have been studying, and we were able to file a number of patents. And the patent with curcumin was again the technology was again used for small molecules, especially in case of rifampicin and anti-cancer molecules like cisplatin and many more. Okay, now briefly let us look into nanotherapeutics. We all know that nanotechnology can be defined as understanding and control of matter at a particle size from 1 to 100 nanometer, where the particles have a unique phenomena, which is not seen when it is either in solution or when it is in larger in size, in the micro range. So this unique phenomena, it enables novel application. So in nanomedicine is the medical application of nanotechnology. It can be used either in therapy or in diagnosis. In therapy, in treatment, nanotherapeutics or nanotechnology can effectively deliver drugs. It can deliver vaccines, and now it is being used to deliver genetic materials. So one example of vaccine is what we have seen, the mRNA. So mRNA vaccine, they have used the nanotechnology in order to see that mRNA is the mRNA can be administered. And in diagnosis, nanotechnology has been used for rapid detection of biological conditions, both in vitro and in vivo. And in the last few years, especially last few decades, we have a number of drugs. It is said that there are nearly 50 molecules being formulated as nanoparticles or nanoformulations. So there is rapid increase in the number of nano products that is there in the market. And it is said that the nanomedicine market is anticipated to reach nearly about 350 billion by 2025. And it has been increasing, and now it is expected to be even more. Now, the reason is because of enhanced efficacy, because of specificity. Now, specificity need not be only, like it need not be actually actual targeting. It can also be passively, the drug can be delivered to a disease site. Then there is reduced toxicity because there is increased specificity, and it is possible to administer 2 drugs. And we can have properties such as sustained release of the drug so that you can prolong the duration of action, especially when it is given as an injection. And then there is rapid and sensitive detection in case of diagnostics. Now let us see why the nanomedicine is actually catching up, why we are having more and more products based on nanotechnology. Now, we will first look at cancer therapy, and then we will try to correlate it with brain drug delivery. So if you see in case of nanomedicine, we have a hypothesis that is the enhanced permeability and retention effect. So what is believed is whenever there is tumor, the blood vessels become leaky. So there are gaps between the blood vessels because these tumor cells are growing at a very fast rate. So since they're growing at a very fast rate, there is no enough time for the blood vessels to be actually compactly formed. So we are having leaky blood vessels. And also there is poor lymphatic drainage because you're having a mass of cells being produced at a rapid rate. So the lymphatic drainage also gets affected. So any drug molecule that enters in, it will be able to be actually present there for a longer period of time. So when we are making nanoformulation, these nanoparticles are able to go through this leaky blood vessel. So here we can see a gap. So it can go through this gap and enter into, through this, it can enter into the tumor site, one. And then it remains there because we are having poor lymphatic drainage. So this is passively we are targeting the drug. We have a number of molecules. For example, paclitaxel is there. Then we are having so many of the drug molecules which are bound to like albumin. For example, we are having vincristine. So many drug molecules have been actually converted into nanoparticles, and we are able to have passive targeting. Now, there is one disadvantage here that once that nanoformulation or nanoparticle gets into the tumor cell, it may not actually release the drug. It may sit there. So the next option is to actually, if you can actively target the drug to the cancer cell. So actually what they do is the drug delivery system, the nanoformulation has certain ligands which are attached, which can bind to the surface receptors. So we are having the surface receptors. So the ligands bind to the surface receptors, they get into the cell, and then they release the drug. So this is another approach that is being evaluated, though we don't have products. But this is an area of active research where they're actively trying to target to the tumor cell. But we have a number of products of passive targeting because it is easy, because the nano formulation can go through the blood vessels, enter into the disease site, remain there for a longer period of time, and because the nano size, they cannot get into the healthy cells. It cannot get into, because they can't bypass the blood vessel. So since they cannot, because of the compact structure, so they can only go through where there is a gap. So as a result, we can reduce toxicity. And it has been found to really revolutionize anti-cancer treatment. If you see nowadays, we are having more and more success with anti-cancer treatment, giving the nanoparticles. We are having albumin-bound paclitaxel, which is being very widely used. So next, now let us try to see if we can correlate it with the blood-brain barrier that is being affected in case of Alzheimer's disease. So if you see in case of Alzheimer's disease, we can see there's a marked change in the structure of the blood vessel in Alzheimer's disease. So you can see that there is decreased tight junction, then altered paracellular pathway, leukocyte infiltration, pericyte is affected, activated microglia, so demyelination of neurons. So all this will result in, again, a leaky capillary network in the brain. So thus nanoparticles can enter into the into the brain because of this leaky structure, sparing the healthy cells, because healthy cells have the tight junction. The structure does not allow the nanoformulation to enter into the healthy site, whichever part may be of the body it may be. So thus we can target the drug to the to the brain in case of Alzheimer's disease. So this is a hypothesis that has been actually being evaluated for drug delivery by nanotechnology for treating these neurodegenerative diseases. Next slide, please. So we have a number of nanotechnologies. We have been, lot of work is going on, but are they commercially viable? So which are the commercially viable nanotechnologies that are being used currently to manufacture, to produce, and to develop drugs, therapies? So if you see the different technologies, we have micelles, which is now a very widely adopted technology to formulate drug as nanoparticles, not only drugs, also for delivery of nucleic acids. Then we have nanocrystals being used. Nanocrystals, the small size of the drug. Then we have these complexes, like as we discussed, cyclodextrin complexes. And then we have liposomes. So liposomes, again, these are the technologies that are commercially viable. And as a result, we have a number of products being released into the market. So I would like to discuss more about this phospholipid drug complex micelles. So this, as a name indicates, we are having phospholipid. So this phospholipid, it is, as we all know, it is a major component of mammalian cell membrane. And it is predominantly, we have phosphatidylcholine, phosphatidylinositol, and ethanol amine that is present. And the major source of phospholipid is, we have soya lecithin and egg lecithin, which is being used. And if you see the structure of phospholipid, so you are having two portions. So you are having a non-polar portion and you are having a polar portion where we are having this phosphate group present. So essentially it is a glycerol molecule. So you are having two non-polar tails and you are having the, you are having one carbon is attached to phosphate group and then it is attached to different moieties. So thus, in other words, it is like a surfactant. It is an amphiphilic molecule. So you are having a hydrophobic portion and a hydrophilic portion. So this has advantages. So our membrane, so we have a bimolecular lipid membrane because of this phospholipid. So it is able to have that is within the membrane, we are able to have a hydrophobic region, and the hydrophilic portion, that is, it faces the tissue fluid, or it faces the core of the cells. So this amphiphilic molecule, it has number of advantages. It is biodegradable, it is biocompatible, it can enhance solubility because it is a surfactant, natural surfactant. Because it is enhancing solubility, it can increase bioavailability of those drugs. That is the class 2, BCS class 2 drugs. That is those drugs which have good permeability but which have poor solubility. And also it is helpful in case of class 4 drugs, like for example, curcumin. And by selecting the phospholipid, we can also retard the drug release. Suppose we are choosing a very hydrophobic lipid, we can also have sustained release of the drug. And it has been said that it gets absorbed. This phospholipid, that is, when it forms a complex, it is because it is a lipid, like how in our diet, when we have a lipid diet, so we have chylomicrons being formed, the chylomicrons get absorbed, they enter into lymphatic system, and then they enter into systemic circulation. So we have lymphatic absorption. So thereby it can avoid first-pass metabolism of drugs. And also by forming a complex using phospholipid, we can mask the taste of drugs. Okay, now this phospholipid has been used in order to prevent, prevent degradation of drugs as well, in order to increase the stability of the drugs as well. Next slide, please. Now this drug phospholipid complex technology. So what happens is, when you are adding phospholipids to water, it immediately forms a micelle. So you are having the hydrophilic head facing the aqueous environment, and in the center, we are having an hydrophobic core. So this is like, for example, if you see that if you are adding phospholipids to a greater, the concentration increases, it again forms a liposome. In liposome, you have two layers with that is the hydrophobic region facing each other. But when the concentration is less, what happens is it immediately forms a micelle, just like soap. When you add soap into water, it forms a micelle, and all the dirt gets into the core, which is the hydrophobic portion. So here, similarly, the drug, what it does is it forms, it gets into the center of this micelle because it forms a complex with this phospholipid. So first we have the formation of a micelle, and then this drug phospholipid complex. The drug will complex with the polar portion of the phospholipid, and thus it gets entrapped within the core. So it is not just getting accumulated. There is weak van der Waals forces, hydrophobic interaction, hydrogen bonding, because of which the drug is present in the core. And this, as we discussed, it is held by hydrogen bonding, van der Waals, and hydrophobic interactions. So as a result of which, that is, the hydrophobic drug gets solubilized. It gets solubilized, it gets uniformly dispersed in the aqueous phase. So when it gets uniformly dispersed, there is increase in absorption of the drug, especially if the drug is having a good, that is poor solubility, and it is having a good permeability. So this technology was developed in 1989 by an Italian pharmaceutical company, and they developed this, they called it as phytosome because they worked with curcumin. And since curcumin is from plant origin, they called it as phytosome. And when we started working with small molecules, we started calling it as pharmacosome. So essentially the principle is the drug, depending on the functional group like COH group, NH2 group, OH group, it forms the bond with the hydrophilic site of the phospholipid, and then it gets into the micelle and it gets held, and thus its bioavailability gets increased if it is given orally, and if it is given as an injection, there is increase in that is circulation time, and also it can enter into the tumor site because of passive targeting. So, if you just to summarize this phospholipid drug complex, it modifies the physicochemical properties of the drug. It can make the drug more hydrophilic, lipophilic. It can increase the solubility and oral bioavailability of drugs. We started with curcumin, then rifampicin, tamoxifen, and other anti-cancer drugs. Then it can increase also the lipophilicity of drugs by selecting the phospholipid. And we can not only increase oral bioavailability, we can also prolong the circulation time by, that is parenteral injection. And the most important advantage is this technology as such is commercially viable. It is a simple technology. So you take the phospholipid and the drug. That is, the phospholipid is dissolved in water. Drug is taken in another maybe organic solvent, depending on the solubility of drug. When they're mixed together, the micelles are spontaneously formed, inside which the drug is entrapped. So that is one of the main advantage of this particular technology. See, the similar technology in case of liposome. You must have studied that liposome. There's a little more additional steps required, where you make a thin layer of the lipid and then you hydrate it with a drug solution, or maybe you hydrate, depending on the solubility of the drug, either you take it with the lipid portion or you take it with the aqueous portion. Whereas here it is a very simple process. You're taking the phospholipid in water, you're taking the drug in a compatible solvent, you're mixing both, and then it can be again concentrated, and then again it can be modified depending on the properties that we require. So here I have just tried to sort of this is actually taken from APS Pharmsci Tech. Here we let us try to see what happens normally to drug molecules. If there is a problem with the drug molecule with like poor solubility or poor permeability, or it is undergoing GI metabolism, it is affected by the enzymes present in the GIT, then you are having, or if there is an efflux, for example, curcumin, there is PGP efflux, then there is very little amount of drug that enters into systemic circulation. But in case of phospholipid complex, this diacyl phospholipid complex, so it undergoes hydrolysis in the stomach and in the duodenum, and it releases these fatty acids, which trigger this cholecystokinin release. And this forms, along with bile salts, you have the formation of the chylomicrons. So this increases the solubility. In other sense, it increases the dispersibility and improves the protection in the GIT. And then there is improved permeation through the biological membrane because it is being coated with a layer of a lipid. So it enters through the intestinal cells, and the chylomicrons, they bypass the hepatic metabolism, enter into a lymphatic system, and then they enter into systemic metabolism. So in other words, we are trying to mimic chylomicrons. So by phospholipid technology, we are actually essentially making something similar to chylomicrons, and thus they enter into the systemic absorption, bypassing first-pass metabolism or pre-systemic metabolism. So this is for GI, in case of, in the GI tract, in case of oral absorption. Now let us look at an example for parenteral administration. So this is an example of patisiran, actually called as Onpatro. So here it is trying to deliver small interfering RNA. So this RNA is for treating transthyretin-induced amyloidosis. That is a genetic disease where there is large amount of this transthyretin being produced. So this transthyretin is again, that is, it causes deposition of amyloid protein in the liver. Like in case of AD, we see that it is in the brain cells, but here it is in the liver cells. So here they try to block the gene itself by using small interfering RNA. Now here, this complicated structure, if you see here, what we have in red is this small interfering RNA, siRNA. And this RNA is held with the help of this phospholipid. And this phospholipid you can see like blue with red head. So this is actually a cationic phospholipid. So it binds to the siRNA because of the anionic charge of the nucleic acids. And it holds the siRNA in place. And in the center, we are having neutral phospholipids and we are having cholesterol molecule. So all this give stability to the this is actually essentially a liposome. So in the center, we are having the core, which imparts stability. So we are having neutral lipids, we are having cholesterol in the core, and we are having charged phospholipid holding the siRNA. And at the top, we are having pegylated phospholipid. Now, this pegylated phospholipid, what it does is, pegylation, because pegylation, as we know, is polyethylene glycol. So polyethylene glycol, it imparts hydrophilicity. And when polyethylene molecule is present around the liposome, the molecule, it sort of, it is like it can evade the immune system. Our immune system is not able to understand that this is foreign because of this hydrophilic moiety. So as a result of which, it protects the liposome from degradation by the enzymes that are present in the blood or in the muscle if it is an intramuscular injection. So thus this siRNA could be effectively delivered. And this siRNA was able to take care of this genetic disease. Now, this was in around 2019, around that time. Now, the success of delivery of siRNA, this led to using this particular approach for delivery of other the genetic diseases. In particularly, it was used for development of mRNA vaccine. So the mRNA, so this siRNA is replaced with mRNA. And so this mRNA is being stabilized within this liposome. And in case of mRNA vaccine for COVID, so this the mRNA is for the envelope of the COVID glycoprotein, the envelope that is present. And this is given by the intramuscular route. So in the muscle, it is able to act as a depot. And this mRNA is released, body produces antibodies, and thus we have immunity against COVID. So this approach was used for production of COVID vaccine. Along with the COVID vaccine, again, this has become a hot area of research. So this particular approach of using phospholipids for delivery of genetic material is now extensively being studied. Okay, so with this discussion, now let us look into look into some curcumin nanoformulations. Next slide, please. So curcumin nanoformulations, I would like to first discuss about a phospholipid that is curcumin complex, which has been again stabilized by a patented technology, followed briefly by cyclodextrins. So let us look at this technology. So we developed this novel curcumin lipid nanoparticles, which exhibited increased oral bioavailability. And this, that is here, what we did is we prepared first curcumin phospholipid complex. So we first prepared that, and then we stabilized it, and then we embedded it in a lipid matrix. So that means we have preparation of curcumin phospholipids, and that was stabilized, and that it was embedded in a lipid matrix. So we observed increase in oral bioavailability. Some of the features of our study include, next slide please. So this is the method. Here again, we adopted the same technology, phospholipid curcumin complex technology, and then we formulated a solid lipid nanoparticle. So the average size of this lipid nanoparticle was around 630 nanomicrons. And so here we have the TEM image. So if you observe carefully, you can see that there is a lipid matrix inside which we are having this phospholipid curcumin complex. So this not only increases the permeability, not only increases the oral bioavailability, it is also able to provide sustained release of curcumin for a long period of time. So then we studied the lipid nanoparticles for its, next slide please. So in vitro release studies. So here you can see that the curcumin, that is the complex converted into a nanoparticle. It had it had increase in release of the curcumin molecule. And also we found that there was increase in permeation of curcumin. So here we use a rat abdominal skin. And so here we are actually comparing two. That is, one, we are comparing the complex with the solid lipid nanoparticles. So here you are actually having pure curcumin, and then you are having the complex, and then you are also having the nanocurcumin. So we can see that here there is increased permeability of the nanocurcumin, whereas pure drug, it could not be detected by the method that we used for evaluation. Next slide, please. Then averted sac technique, we found that there was enhanced permeation or curcumin from nanocurcumin, and bioavailability studies also showed that there was considerable enhancement in the oral bioavailability of curcumin. Again, here what we saw that was the complex definitely increased the bioavailability of curcumin, but the pure drug, it was again undetectable by the method of analysis that we used for the study. So we can see that there is a considerable enhancement in the bioavailability of curcumin. Then we studied the efficacy studies. We had number of that we found that it was exhibiting increased anti-inflammatory activity, maybe because of increased permeation, larger amount of curcumin being present. And as Professor Sharma said, we studied its activity and using that diabetic neuropathic model. And again, we saw considerable enhancement in the activity. Next slide, please. So thus our patented nanocurcumin, it exhibited prolonged and enhanced oral bioavailability. And this could be attributed to enhancement and prolonged in vitro release of the drug. It was released for a longer period of time, increased permeability, and increase in the oral absorption of curcumin. And thus we can say that nanocurcumin holds enormous potential in prevention and treatment of Alzheimer's disease. And this therapeutic potential could be used either alone or in combination with currently used drugs, or maybe the drugs that are going to be used in future. So this is one formulation that I wanted to discuss. The other, briefly, I wanted to look into cyclodextrin complexes. Next slide, please. So this we worked considerably on this curcumin cyclodextrin complexes. We work with alpha CD, we work with alpha cyclodextrin, beta cyclodextrin, gamma cyclodextrin. So this cyclodextrin, they are formed from starch by action of a microorganism, Bacillus messerins, and it forms a cyclic structure. So this cyclic structure, depending on the number of molecules that are present, this cavity size, it changes. So here, the core of the cyclodextrin is hydrophobic in nature, whereas the OH group of starch molecule, they're all present towards the exterior. They're present towards water. So as a result, they increase the water solubility. And all the hydrophobic drugs, they can get into this hydrophobic cavity and form an inclusion complex. And this inclusion complex can be formed in a very simple manner. That is, it can be, the preparation again is by simple methods of forming a complex, by say common solvent method, pulverization method, spray drying method. So all these methods which we commonly use in pharmaceutical technology can be used for preparing the cyclodextrin complexes. So here we have alpha CD, beta and gamma. Now we worked on beta CD. Then one company, a Japanese company, wanted us to work with alpha CD because of the molecular size of alpha CD. So if you see the size, hydrophobic cavity is less. And if you look at the molecular weight of curcumin, it is only 368 Daltons. So it can be incorporated easily and held within the alpha CD molecule. So we prepared the cyclodextrin complex by pulverization method. We also tried the spray drying method. And alpha CD has considerable amount of solubility when compared to beta. Again, gamma is also having good solubility. So we prepared the cyclodextrin complex and we observed, again, increase in solubility and increase in oral bioavailability of the drug. Next slide, please. So we found that when we took curcumin, alpha CD showed a considerable enhancement in the solubility, followed by beta and gamma CD, and this could be attributed to the hydrophobic interaction between the drug, between curcumin and the CD, the hydrophobic cavity of the alpha cyclodextrin. And here we have the dissolution study. So in the dissolution study, we can see that alpha CD, it has the enhanced dissolution rate. And dissolution rate can again help in increasing the oral absorption because it will saturate the enzymes that are present in the GIT so that more amount of drug can enter into a systemic circulation. And since the dissolution solubility was increased, we found that there was an increase in the oral bioavailability too. Next slide, please. So we can see here that alpha CD showed considerable increase in the oral bioavailability of curcumin. So pure curcumin, you can see that there is a significant increase in the bioavailability of curcumin when compared to beta and when compared to gamma. So this could translate again to an increase in activity for neurodegenerative diseases in general and Alzheimer's disease in particular. Next slide, please. So with this, I would like to conclude that nanotechnology holds the promise of enhancing the therapeutic potential of curcumin in preventing and as well as treating Alzheimer's disease. And the curcumin nanoformulations, they have the potential to optimize AD therapy either alone or in combination with current therapy, especially to benefit advanced AD patients. So with this, I thank you all for your kind attention. Thank you.
Thank you very much, ma'am, for your valuable information and valuable talk. Now the session is open for discussion. If anyone want to ask any question or discuss anything, you are open to ask.
Yes, ma'am. Very good afternoon, ma'am. Ma'am, I want to ask what is the best particle size for brain delivery or nose to brain delivery in nanometer or micrometer? Some researchers says nanometer size is best, and some researchers says micrometer size best. Some researchers says mucociliary clearance does not allow micrometer, does not allow nanometer. Matlab, ma'am, what is the best criteria for nose to brain delivery for Alzheimer's disease?
See, actually, strictly speaking, if you're looking at nanotechnology, it says that below 100 nanometer, the particles are exhibiting a particular property that is which helps it, say, to be either, to cross the nasal barrier and to enter into the into the brain. But it has been found that even above 100 nanometers, studies have shown that. See, for example, in our studies, it was around 600 nanometers. And in many researchers have reported that an increase of 200 nanometers also increases the amount of drug that is being delivered based on the composition of the nanoformulation. So it is, we cannot actually, it depends also on the characteristic of the nanoformulations that we are using in order to deliver the drug. So what is the type of polymer? What is whether it is biocompatible? So they all seem to play. That is what I have found out from the literature. So there is no such a, like, you cannot just rigidly say that this particular size will be really be beneficial. See, for example, if you see nasal vaccines of COVID, they are being developed. They're also adopting the same technology. So the particle size is the same. So you must have seen that now the products, there are a lot of research is being done for nasal delivery in COVID vaccine. So I think it depends on the formulation also.
Yes, ma'am. Thank you, ma'am.
Okay, thank you. Anyone else?
I have a quick question.
Okay.
So thank you, ma'am, first of all, for sharing your wonderful research. And my question is about, specific about nanocurcumin and the effect on Alzheimer's disease. So did you ever check about the efficacy of these nanoparticles both in males and females?
I have not studied, in Alzheimer's model What we were looking at is, we are looking at, in case of neuropathy pain in diabetes neuropathy, we did study. So neuropathic pain, we found definitely nanocurcumin was able to alleviate the pain that is associated with diabetic neuropathy, definitely. But we have not actually gone down to the molecular mechanisms per se. But since that is why I was correlating it with, enhanced permeability and retention effect. So since there is, in case of all these neurodegenerative diseases, the blood-brain barrier gets affected. So the chances of drugs entering into the nanocurcumin entering into the brain is, increased.
So that's my question about, because since my research is all focused upon the sex differences in the brain, basically what are the differences in males and females, and now a lot of researchers are like compelling us to just use both the sexes whenever you are designing any kind of studies like this. And as far as my research is concerned, so we have observed like higher blood-brain permeability in males as compared to the females. So that might have some kind of a connection between some drugs have effects on males and some have effects on females. So I think this is the time to go for the precision medicine. So we can manage our formulations and drug types and concentrations and timings and so on.
So you mean to say, okay, the sex of the animal is also having a role to play. That is what you are trying to say.
I'm trying to say the sex is the major factors that again playing in this like efficacy of most of the drugs.
Okay.
As far as the neurological disorders are concerned.
Okay. Good information. Thank you.
Thank you. Thank you.
Thank you very much. So anyone else?
Good afternoon, ma'am. Shachivarli this side.
Good afternoon.
First of all, thank you so much for such an informative session, ma'am. We are actually the lucky ones because we got this opportunity. Ma'am, actually, I have worked with the pipeline on neurological disorders. So I studied bit about the curcumin also, and I found that polysaccharide complexes are able to easily cross the blood-brain barrier. So my curious question is curcumin loaded phospholipid micelles or phospholipid complexes like lipospheres, liposomes, are they also able to cross the blood-brain barrier easily with? And I'm also curious to know about how they cross the blood-brain barrier, and is there any mechanism we follow or something like this? So I would like to know about this, ma'am.
So we could see, again, it is a hypothesis. We all know that even the enhanced permeation retention factor, that is also a hypothesis. So it could be that since it is leaky, we saw that parallel cellular pathway, it is leaky. So through that, like how the immune cells enter in AD, same way the nanoparticles could enter, nanoformulations could enter. Now, if you ask me what is the advantage of this phospholipid complex, is that fact that phospholipids, they're biocompatible. There's something that is the toxicity with respect to phospholipid complex is definitely less when compared to some of the polymers that we are using. So if you see that is one of the criteria why we are having now nucleic acids being delivered by this micellar approach, because of the biocompatibility and safety of this phospholipid. So I could say, and also another advantage, if you see the method that has been used in the preparation of COVID vaccine, mRNA vaccine, again, it is just simple mixture. Of course, you have to optimize all the components, you have to optimize the rate, the equipment, but it is simple mixture of the phospholipid solution and the mRNA. But so it is just a admixture. And this phospholipid is formed, and the lipids, they spontaneously form the liposomes. So I would say that phospholipid, because of its biocompatibility, safety, and ease of preparation, it does have an advantage.
Thank you so much, ma'am. Like we can say that how the Trojan horse process works in the case of dopamine when it crosses the blood-brain barrier. The same way our phospholipid complexes are working as the Trojan horse model to incorporate, like when we incorporating our curcumin or any other molecule into it. So it is working like that way to reach the target.
Right, yeah. It gets into, and then lysosome breaks that, and then it is released, and then it brings about the therapy.
Thank you so much.
Thank you very much. Anyone else?
Ma'am, one question more, please. Ma'am, जैसे कि मैम कर्क्यूमिन है, कर्क्यूमिन को मैम जैसे हमको एल्जाइमर के लिए हम उसको ट्रीट करने के लिए दे रहे हैं और हमने उसका नैनो फॉर्मुलेशन बनाया, ठीक है, और मैम हमने उसको सपोज फॉर इंटरनेजल डिलीवरी या मैम अदर डिलीवरी हमने उसको दिया, तो मैम देयर आर मैनी पाथवे, ओल्फैक्टरी, ट्राइजेमाइनल, देयर आर मैनी पाथवे दैट मैम व्हाट इज मतलब मैम इट इज नॉट नेसेसरी कि दैट कि हमारी जो फॉर्मुलेशन है वो डायरेक्टली ब्रेन तक जाए, उसका कुछ पार्ट हो सकता है मैम किसी अदर पाथवे में जाके, जैसे ट्राइजेमाइनल और अदर पाथवे में जाके हमारे ब्लड सर्कुलेशन में भी जा सकता है और हमारी अदर ऑर्गन्स अदर पार्ट्स में भी जा सकता है। तो मैम ऐसे भी हो सकता है कि ब्रेन पे कम इफेक्ट हो, वहां ज्यादा चला जाए या वहां ज्यादा चला जाए, कम चला जाए, तो मैम क्या हमारी बॉडी में जाके उसके साइड इफेक्ट्स हो सकते हैं नैनो फॉर्मुलेशंस के फॉर्म में?
See, yeah, it is definitely true. Like it can enter into the systemic circulation, but again, we have to remember that, you know, I mean, this is my, I'm just trying to think. So one could be that, you know, you're having curcumin, which is again definitely not applicable. You're having phospholipid polymers, which are biocompatible, safe. That is one point. Second point is this nanoparticle, when it enters into the systemic circulation, because of the nano size, it is not able to reach, enter into healthy tissue. So it enters into tissue to a greater extent where it is not, you know, where it is not compact. The cells are not compact. So through the that is systemic circulation, definitely I believe that it can be targeted to the brain. Just like how it is parenteral. Parenteral also, you're injecting it into systemic circulation, then it enters. In case of chylomicrons, we believe that, you know, it is having the phospholipid, it is in the form of micelle, which gets targeted to the brain. So again, because of the leaky nature of the blood-brain, there could be some amount of targeting by systemic circulation too.
Yes, ma'am. Thank you very much, ma'am.
My pleasure.
Okay, thank you very much.
Ma'am, one quick question.
Okay.
So, is your formulation is commercially available right now?
We patented. Yeah, we have some companies that approached us. Yeah, so we are. I mean, it is still in the discussion stage. Yeah.
Okay. Thank you.
Okay, thank you very much, ma'am. So thank you very much, ma'am, for providing us the valuable insight on the potentials of nanocurcumin for optimization of Alzheimer's therapy. So we greatly appreciate your time and effort, ma'am. So please accept our sincere thanks once again for your precious time. Thank you very much.
Thank you so much. Thank you all. It was a great pleasure seeing you, Arti, after a long time.
Same here.